Hello, I'm Koji Matsura and will be a professor in the medical biology course. I will explain what you will learn in this course. You will handle animal tissues and cells prepared in vitro. You will also study new technology relating to future and regenerative medicine. The yellow line is not lightning, but shows a way directing to your future. I have a membrane for blood dialysis with my right hand. The device attached to my left finger is pulse oximeter. Using this device, you can analyze blood oxygenation at home. These medical devices are prepared based on engineering. Studies to cure disease by regenerating tissues advance using potential of culture cells using such as iPS cells about 10 years ago. At present, the medical trials using the cells start in Japan. Stem cells have a potential to differentiate into cells for each organ. When we collect cells in vitro culture and aggregate them, a tissue model of airway or capillary blood vessel can be prepared in vitro as shown in this image. Implantation of an in vitro developed tissue is considered more effective than the cell implantation. Thus, throughout your learning in this course, you can master handling stem cells and regenerative medical technology based on tissue engineering. Next, you will check structures and function of proteins smaller than cells. This image is a molecular modeling of hemoglobin. This protein has a heme and dioxin can bind to an iron molecules of the heme at the center of this image. Cells' functions are controlled by biomolecules including proteins. You will understand protein functions and technologies for developing an apheresis device using protein-protein interaction as shown in this image. These subjects are also important in this course. New tools have been developed for cell function modulation and analysis. You will learn nanotechnologies as a tool for new medical technologies. For example, this image shows functionalized liposomes in nanoscale can encapsulate double-stranded small RNA and RNA interference mechanism to knock down. One of our studies includes the optimization of this function. This slide shows some instruments and experiments. The girl in upper left image manipulates cells in a clean bench. You must handle the cells in a clean environment like in the clean bench. Right image shows an optical absorption analysis using 96 well plastic plate. Upper left and the lower right image shows DNA analysis and application respectively. You will learn this experimental skill in medical biology course, as the same as in other courses. This slide shows future technologies of treatment and diagnosis. So far, instruments and methods for medical technologies have been developed based on engineering. In future, cell function will be used for development, and this course will contribute to research and development. Thus, you will contribute to improvement in tissue reconstruction, treatment, and diagnosis efficiencies and personal medicine, which are not realized now. After graduation from this course, we intend you will work at a pharmaceutical company, a company providing medical devices, a hospital, and so on. Through lectures and studies in laboratories, we provide your skills for future technology developments for treatment and diagnosis in order to foster your ability to be a main player for the developments. We are awaiting you at the medical biology course. See you.